Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Brooker-Anthony, Scientific Editor of the Molecular Therapy family of journals. This episode features a discussion led by Dr. Paloma Giangrande, Editor-in-Chief of Molecular Therapy Nucleic Acids and President and Founder of Biologic Insights, LLC, with Dr. Ayşegül Yildiz, Scientist of TRON's Cardiovascular Therapeutics Group, and Dr. Tim Beissert, Senior Scientist and Group Leader of TRON's Vector Development and Gene Transfer Team. They will discuss a recent article published in Molecular Therapy Nucleic Acids by Drs. Yildiz, Beissert, and colleagues titled Trans-amplifying RNA expressing functional miRNA mediates target gene suppression and simultaneous transgene expression. Are you registered for ASGCT's 2025 annual meeting in New Orleans? Meet us in New Orleans, May 13th through 17th, for the premier event in our field and save $465 by joining ASGCT and using the incredible discounts the Society provides to members. Check out the full program, plan your trip, and register for the ASGCT annual meeting by visiting annualmeeting.asgct.org. And now, Dr. Giangrande, Yildiz, and Beissert.
Great. So today we're diving into the exciting world of gene therapy and gene regulation. Our focus will be on a groundbreaking approach to co-delivering microRNAs and protein coding RNAs, opening new doors for therapeutic applications where protein restoration or downregulation alone is not sufficient to achieve a therapeutic effect. So imagine combining gene expression and gene regulation into one powerful package. Sounds very intriguing, right? Well, that's exactly what our guests, Dr. Beissert and Dr. Yildiz, in collaboration with researchers at BioNTech, are exploring with their trans-amplifying RNA technology, platform technology, or taRNA platform technology. So without further ado, let's delve right into this fascinating platform. Dr. Beissert and Dr. Yildiz, it's a pleasure to have you here today. Maybe we start with you, Dr. Beissert. Can you outline for our listeners the concept of trans-amplifying RNA and how it differs from traditional RNA delivery methods or for canonical mRNA or microRNA therapies?
Yeah, thank you very much for inviting us to this podcast. Yeah, sure, I can do that. So the trans-amplifying RNA is a RNA that combines both self-amplifying RNA and classical mRNA. So we have both of these types of molecules in the platform. mRNA, as you know, is primarily for protein translation and used as such in therapies. You have a very high translation, but also a rapid degradation of the RNA after delivery. There have been really considerable improvements regarding RNA stability, but still it's degraded. On the other hand, you have self-amplifying RNA, which is also quite known in the meantime, where there's on the mRNA a second open reading frame that encodes for an RNA-dependent RNA polymerase, so that you get a transient amplification of the RNA by transcription of new molecules, and by that a longer and a higher expression level. But it's a very long RNA molecule, and this replication of this RNA is for cells is a burden for the cells. So what we have done is to combine both of these molecules. So we took the RNA polymerase onto an mRNA molecule, which is not amplifying. So that's a canonical mRNA, having the same design as used in the Comirnaty vaccine, for instance. And what is amplifying is a second molecule which we provide in trans to this. So it's a co-transfection method. And the replicase acts uniquely on this second RNA. And we also call it transreplicon for this reason. And it's very short. It's in our current version, it's looking like an mRNA, basically, but the 5' and 3' ends are virus-derived so that they can be amplified by the polymerase. So the whole system of self-amplifying RNA and trans-amplifying RNA derives from alpha virus genomes, and also the enzymes behind are all alpha virus enzymes. That is the main difference. We have a short amplifying RNA, and we found also that this is, for instance, less toxic to the cells. So because we think that because there are less RNA mass that needs to be amplified in the cell, so you need less energy, less nucleotides, etc. from the cells. So that's why we think it is for the cells less toxic.
Yeah, that's great. Sounds very exciting. So, and with respect to the microRNA aspect of the platform, is this similar to other microRNA platforms that have been described before, or are there several advantages of yours versus what has been described?
So first of all, thank you very much also from my side for the invitation. It's a pleasure to be here. And maybe I can start with saying, because this was a project in the frame of my PhD work, and the intention was not to create a new microRNA delivery platform, but rather address the question if our platform that is made for protein expression can also simultaneously produce microRNAs. And maybe also a bit storytelling here. So there were several papers almost more than a decade ago now that showed that you can engineer RNA viruses in a way that you can incorporate microRNAs, and these RNA viruses indeed can release microRNAs within the cytoplasm. And that was actually the hint for us, let's try it with taRNA and saRNA, because it was not clear if it will work. But it worked, and the surprising thing was that we were not seeing any reduction in transgene expression. So that's where the potential came in. Like, it has a potential because if you think of microRNA delivery, we don't think of our platform because our platform is not the most efficient one for microRNA delivery. There are more efficient platforms for sure, like such as just using siRNA or shRNA, or for stable expression, lentiviral vectors. But the combination basically makes this platform attractive.
Yeah, that's great. So it seems like you're not really reinventing the wheel there. You're just taking something that's already been de-risked and optimized even in the clinic, and you're applying that to your platform now to essentially kill two birds with one stone. So you're taking platforms that have already been de-risked, putting them together and making them work even better together, which is really, really fascinating. So, in your opinion, what are the potential advantages, which I think we've already covered some of these here, of using the taRNA platform for delivering both protein coding RNAs as well as microRNAs simultaneously? Whoever wants to take this question.
As Aisha just mentioned, the taRNA platform was initially primarily for protein expression, a very strong protein expression, and it's a vaccine vector platform as we see primarily. Of course, incorporating therapeutic proteins that act in cells are also facing obstacles. So you might want to use a therapeutic protein, and you realize that there are other host proteins maybe in the cells which counteract the therapeutic protein. And so having a microRNA on the same molecule would forcibly deliver the microRNA to the same cell where the therapeutic protein would act. If you know a target, you could target this inhibitory protein with a microRNA. That's one of the advantages that we see. You could also Yeah, it's the cherry on the cake, as we say. So you have your therapeutic protein, and if you know, in addition, a microRNA, you could use it together. For instance, we think it could be used, for instance, to dampen maybe the innate immune response to the RNA, because this is a big issue. If we deliver therapeutic RNAs, innate immune responses are activated, and if you could design a microRNA that acts against this could be beneficial.
Yeah, that's fascinating. So any existing mRNA therapy out there that has these potential immune side effects, your technology could potentially improve on that by suppressing some of these pathways. Yeah, that's really fascinating. In addition to that, are there indications that really lend themselves to this co-delivery or combination strategy that you can comment on?
Sorry, I think I did not really understand this question fully.
Oh yeah, like for example, indications where there is like a phenotypic, both toxic gain of function, for example, as well as a loss of function. For example, one of these diseases that I've worked myself on in the past is alpha-1 antitrypsin deficiency, where in the liver it presents itself as a toxic gain of function, which would really lend itself to your microRNA sort of or siRNA mechanism. And in the lung, it presents itself with a loss of function. So then, replacing the lost protein or the lost gene could have a therapeutic impact. So I was wondering if you were aware of other indications where your technology is of a specific niche for these indications that are a specific niche for your technology.
Yeah, so for the moment we are platform providers. So we do not ourselves for the moment. We don't work on a specific application for this combined platform, but we see the potential. So it's rather, I think, for people that are working on diseases that could identify this specific combination, which is also, I think you really need to have a deep knowledge of the diseases too.
So you are encouraging a lot of biotechnology companies to start thinking about where they can apply your technology. Yeah, that's great. So let's move to the next question. So you've demonstrated that that your platform can effectively deliver functional microRNAs as well as protein coding sequences simultaneously. And perhaps somewhat unexpectedly, at least that's my understanding, you observed that the amplification of the transgene or protein coding gene that harbors the microRNA sequence is necessary for achieving target gene regulation and long-term suppression of the microRNA target. Can you say why this was not a trivial observation or necessarily an expected finding, that it was quite actually unexpected, especially given that the subcellular localization of these two mode of actions are quite different. One, the trans-amplification takes place in the cytoplasm, while the other, the primary microRNA processing, is thought primarily to take place in the nucleus. So can you maybe expand a little bit on this and your thoughts on that?
Yeah, for sure. I can answer these questions. These are great questions, and I'm coming back to basically the beginning of our research, where we again found those papers. And actually, those papers gave us indication, actually an RNA virus could be a potential delivery platform for microRNAs, because as you just said, it is not trivial, because we thought that processing of microRNAs always start in the nucleus. And these papers showed, no, it doesn't need to be necessary in the nucleus. It can be also in the cytoplasm. And in one of these papers, they showed that TRON, which is the first endonuclease that is needed for the first processing step of the microRNA, is located in the nucleus, but upon an infection, these nuclear TRON enzymes actually translocate into the cytoplasm. And with that in mind, we said, okay, let's give it a shot. And we saw, okay, there is actually microRNA processing. But then for sure, the next question was, okay, if we have efficient microRNA processing, does that mean that our protein expression will suffer? And we didn't see that. And our hypothesis is why we don't see that is because we have so many copies, like the replicase produces so many copies of the template that the small number of templates that are used for microRNA processing do not have any effect of the transgene expression per se. And I think that is really the reason why this platform has this potential. Because we see with conventional mRNA, actually, we also see microRNA processing. So I detected mature microRNAs in the cells, which were transfected with conventional mRNA, which also encoded a microRNA. But just a couple of microRNAs, and we didn't see any knockdown effect because there were clearly too few microRNAs. So we need this amplification by the replicase to really have an impact in the knockdown efficiency.
And maybe this is another question for you, Dr. Yildiz, and you had touched upon this even earlier. So how did the knockdown efficiency of the taRNA, microRNA, how does that compare to the typical efficiency observed with stable lentiviral microRNA transfer? You had said that it's not as good as that in your previous answer. Do you think this implies that the processing of the cytoplasmic RNA is rather inefficient compared to that of the nuclear processing? What are your thoughts on this?
It's a very nice question. We believe that there are probably several limiting factors that are limiting the increment of the knockdown efficiency. First, for sure, the nuclear localization could be one, that there's just not, let's say, enough TROSHA enzymes in the cytoplasm to process our template in the first place. But it's also known that the microRNA machinery, the RISC complex, that is also there for endogenous microRNAs, compete with, in this case, with our templates. So there's also competition going on in the cell, and it can be very fastly saturated if you just transfect the cells with too much template. There can be a saturation. This can actually cause toxicity. So there's also a limit. So we cannot give too much RNA into the cell. At some point, the cells will just die. And it's transient. No, we have only for a couple of with trans-amplifying RNA for sure, a couple of days. We see the knockdown efficiency. With conventional mRNA, it's even shorter. So it's also a bit unfair to compare our technology with the lentiviral system, because there you have constant expression of the microRNAs. And even with siRNA or with artificial microRNAs, you also get a higher knockdown efficiency. But what I think what our technology has an advantage is that we have a prolonged knockdown efficiency because of the amplification. We see that the knockdown can be extended, which in this case makes definitely an advantage here in the technology.
That's very interesting. So given previous observations that these mature artificial microRNAs that are released during processing can, in theory or theoretically, counteract replication or translation by self-targeting complementary sequences within their own related hairpin structure. How do you explain the regulation of the target by the microRNA that you described did not compromise the stability of your STR microRNAs to be amplified or to express the co-encoded proteins? So how's your system working so well? Like, what are your thoughts on that? How is combining these two, what makes them work so well?
Maybe about the self-targeting. So we discussed this also in the paper. We have, kind of also two hypotheses. So one would be there is no self-targeting at all, because one reason could be that our transreplica, especially the untranslated regions, are highly structured. And this potentially masks the target sequence or shields it from the processing by the microRNAs that are produced. The other one is RNA viruses replicate within membranous compartments, which also naturally are created to isolate and to protect the replication process. And this could also maybe shield the whole self-targeting system. But it could be also that there is actually self-targeting. We don't know. We didn't investigate it deeply in this, but because again, the theory of having so many produced copies, self-targeting that does not quantitatively affect and transgene levels in the first place. Because it's just so much template, it's fine that there are a couple of templates targeted by the microRNAs. So these are our thoughts on it, but we did not investigate in this direction.
Yeah, I guess if the system is working, there's no need to worry about it. Again, and this is one thing I've been curious about, and I haven't asked about yet, but is there a limit to the number and types of microRNA sequences that can be incorporated into the STR?
We cannot answer this with a number because we did not go on to chain so many microRNAs as possible. We chained up to 5 microRNAs and it worked very well.
That's great. Yeah.
I can expect that there could be even more than that. So the UTR can be extended. I guess at some point the length will be maybe an issue because of the stability. And also the repetitive sequences, because of the backbones, they're always the same. That could also make a problem. But one needs to test this. We did not go on and copy 10 or 20 or 30 microRNA copies in one molecule.
But in theory, it's definitely possible, and it's just a technological sort of optimization step that may be required. But you could potentially go after the regulation of multiple genes that could further augment your therapeutic outcome. So I think it's really a really interesting area and future, in my opinion, for this technology. Maybe let's move a little bit and talk a little bit more about the technical challenges and your solutions to some of these challenges. Because reading the paper, obviously, it seems like this was so seamless, and that you woke up one day and managed to develop the system and everything worked great. But in reality, we're all aware that there are a lot of hurdles along the way, and some of these hurdles are not intrinsic or trivial, and there's a lot of work, and especially on your end as part of your PhD training to try to overcome these challenges. So if you can comment on some of these challenges and some of the solutions that you came up with that made this a reality and ended up in an amazing publication in our journal.
Thank you very much. I have to admit, I was very lucky with this project because we didn't have major challenges. It was pure luck, I guess, because with my other project I had several challenges, but with this project everything worked quite smooth. We did not the project ended with the PhD program ending, but we did not dive into the improvements. So that's something we definitely want to do. And I guess there probably we will find some challenges by optimizing the system. But the proof of concept experiments, they just worked very good, I have to say. Tim, I don't know, if you want to add something?
That's great.
Yeah,
a great idea.
It was one of these PhD projects where when you found some publications and you say let's try it, where indeed you could say that what was in the publications obviously was true. So the people have shown in the alpha virus the viral transfer that microRNA could be transferred. So we were pretty sure that it should work because of the RNA replication in these viruses, and for saRNA or also taRNA are so close and so similar, almost identical, so that we really expected it. So if they are right, these people, then it should work. The question was more, will there be enough microRNAs to have an effect? That was rather the question. We were pretty sure that we would get mature microRNA, but the level was completely unknown to us. And also, as we discussed before, would that harm the protein expression? Because not only A potential self-targeting could be a problem, but also you need the right balance. So RNA replication needs the 5' and 3' terminal ends. So if Roger chops up the RNA, this molecule would no longer replicate. So if you would have a very high efficiency, like, I don't know, 95% would for sure stop RNA replication. And if it's too low, we would not see any effect. But obviously, it seems to be naturally quite nicely balanced.
Yeah, and so we've talked a little bit about that self-targeting, which it doesn't seem like it's a main issue right now. Can you comment a little bit on the potential of off-target effects with your approach? Are these any different from what any other technology or similar technologies are facing? And also on the potential of the emergence of unexpected microRNAs from the vector backbone that you have designed?
Well, I think from the side of the microRNA itself, you face the same off-target issues as with other formats.
Yeah. Okay.
So you need to carefully design your microRNA sequence. You need to apply bioinformaticians tools to check if you can foresee any off-target effects. So unexpected microRNAs, of course, yes, they could arise. It has been shown, I think, for flaviviruses that they can derive from the genome. I think the question for a therapy is whether these microRNAs would by chance then target something in the host which would really cause severe side effects. I think so. When using the trans-amplifying system, there's one advantage that the replicase open reading frame, which is very large. So the replicase makes up 8 kb approximately. And we put that on a non-replicating RNA. And by this, we reduce all the potential microRNAs that could arise from this part, because this is not co-amplifying. And so the unexpected microRNAs that come from the gene of interest, of course, this could happen. But here again, I would say using the right tools from bioinformaticians should help to reduce this likelihood that you get harmful microRNAs from these sequences. But this is not our core expertise, so I cannot comment more deeply on this. But I think having really advanced tools now in bioinformatics and artificial intelligence, this all should help to minimize this risk. But it's not specific to our platform, that applies to all microRNA platforms.
Yeah, makes sense. And can you comment a little bit on the delivery system and if this can and other delivery approaches can be applied to your platform? For example, for taking this to specific target tissues, maybe even extrahepatic targeting. Any thoughts on that?
So yeah, that's an advantage of the platform, since we only have one type of RNA. It's a long RNA, so in the range of messenger RNA. So you can use the delivery methods that are now established for mRNA for the vaccines. So lipid nanoparticles can be used. And you do not face the problem that if you think of the co-delivery of these long RNAs with a very short microRNA, so you do not need to take a second formulation for the microRNA. It's just one molecule that all fits into this one LNP, for instance. So we think that any formulation that is developed for long mRNA should be applicable for the platform. Especially especially those that work with self-amplifying RNA should be good candidates.
Yeah, that's very exciting to hear. Definitely opens a lot of doors with all the improvements in the currently undergoing in the LNP space, especially for developing targeted LNPs. So maybe let's move a little bit towards the future development of this technology and potential translation. So this is, more speculative type of questions. In your mind, what are the what do you see as the next steps for further research and development in this area?
So one thing we are currently working on, and we are close to getting this published, is to reduce the immunogenicity of the RNA itself. So as you know, the vaccines have modified RNA, and so far self-amplifying RNA was shown to be inhibited, for instance, by the 1-methylpseudouridine modification. And we've been working on in the past years on adapting the platform to 1-methylpseudouridine modification, and we found that it worked. And we will hopefully soon finish this manuscript. We're also working with viral interferon inhibitors that are proteins that counteract interferon response. That's also mentioned in the paper more as not such a prominent figure maybe, but that was also used in the paper to reduce the interferon response in the primary cells that Ayşegül was using in her project. And we also think that using these inhibitory proteins is also something that can be applied in a therapeutic setting.
And given that this was a collaboration with a biotechnology company, BioNTech, can you share any insights in, with regards to this collaboration and potentially moving forward with them? Any interest that you see in general in taking this platform into the clinic? And how quickly should we expect to see this platform into the clinic, I should say?
I can share a few insights. We cannot go into detail with that. It's a bit sensitive. I hope you understand. So TRON and BioNTech share a common history, so to say, when you go back for 12, 15 years. TRON was founded by Uğur Şahin. He's one of the co-founders. So also other people, also and others have founded TRON and BioNTech. So this is the natural connection between these two companies and institutes. We at TRON, we do a translational research. We are collaborating in many projects with BioNTech. But what we are not manufacturers. So we do not go on to really make products of the findings that we have. So this would be more the industrial partner to do that. So that's the sharing of the work in our collaboration. How long it will take? Yeah, that's a good question. Hopefully not too long, but I cannot comment on this.
I think one thing I would say is given what we were talking about earlier as well, is with respect to the fact that you're taking some technologies that have been somewhat de-risked clinically already, and you're combining them in such a unique way to achieve desired effects for certain applications or going after certain indications. I would think, but I leave this to the experts like BioNTech and others, that both the manufacturing as well as the regulatory process or clinical development process of your technology could be somewhat expedited in moving forward. But still very exciting. And I'm sure, optimizations of the platform are, going to make it even better. But definitely something to look forward to and to keep an eye on. So I'm going to ask you one last question, and this is absolutely very speculative. And a little bit of an interest of mine, given that I am very modality agnostic, and I appreciate, the various niche for various modalities. I think some modalities obviously work better than others and cater themselves to certain areas or applications much better than others. So how do you foresee the incorporation of microRNAs or other regulatory elements, such as instead of microRNAs, but for example, small activating RNAs or RNA editing antisense oligos into your tRNA vector platform? And do you think this is a possibility that you guys are interested in exploring in the futures, or others may explore in the futures? Is there an area for combining some of these other modalities into your platform?
Yeah, I mean, we did not further went through, but of course we had also in mind that they are not only microRNAs, right? There are also other non-coding RNAs that are impactful that can be tried.
Regulatory RNAs, yeah.
Exactly. And I also wanted to mention, I didn't mention this at the beginning, but we also, so this microRNA delivery also works with saRNA. We also did that with saRNA, so
Perfect.
with both technologies it's possible. It needs to be tested. That's the simple answer. What we really want, what I wish to do is really to improve the whole system even more, because what we saw in the paper was the highest knockdown efficiency. I mean, it was only one target we are showing in the paper. It was 80%, and 80% knockdown efficiency is, yeah, when you compare it with other systems, is definitely not so much. Usually it's 95, even higher. So to push more in that direction, so it gets also more attractive for this for the knockdown efficiency. But yeah, I think we have ideas also for application, which we unfortunately cannot talk about today, maybe in the future. But we are also very open for other ideas, as you just mentioned. Yeah, Tim mentioned about different diseases, and your example, definitely something that could be looked into with this platform.
Well, this is very exciting, and I want to thank you both. I think this brings us to the end of today's episode, and we hope you enjoyed the discussion and gained some valuable insights regarding trans-amplifying RNA technology. Again, thanks for listening, and see you next time. Thank you both.
Thank you very
Yes,
much.
thank you very much.